I'm standing here in an experimental plot. This is a collection of 42 accessions of Aegilops tauschii. This is the D-genome ancestor of bread wheat and considered to be a repository to potentially valuable genetic diversity. So we planted these out here at Ardek, north of Fort Collins. We planted these under fully irrigated and under dryland conditions. So one of the traits that we hope to look at is the drought tolerance of these materials. But these were collected everywhere from Syria to China. Quite a few are from Iran, from Armenia, from Afghanistan. And as you can imagine, from that wide range of habitats, there's a lot of different ecotypes of this species. So let me point out a couple right here. This one is from Iran, and look how close it is to the ground. It looks more like crabgrass than it does like wheat. It's just spreading its tillers very close to the ground, whereas this one from Turkmenistan is very upright. It has more of a wheat-like appearance as far as where it holds the seed heads. Another difference is the amount of biomass produced. So biomass we define is everything above ground, the combination of the leaves, the stems, and the seed heads. So this one here, this is from Azerbaijan, and produces a huge amount of biomass. It's just really spread a lot, produced a lot of tillers, tiller being a stem with a seed head connected to it. So a high biomass one right next to one of very low biomass. So not very much leaf or stem material here, and the amount of seeds also very restricted. Here's one that is very prolific. The combination of number of tillers and number of seeds per tiller would make this a very productive line. So what we hope to do here is to pick out maybe 5 or 6 that we consider have some potential, that when crossed into wheat, they would be beneficial, perhaps giving greater biomass or a greater number of tillers with seed heads. It's not an easy thing to do because these accessions only have a single genome. They have the D-genome, whereas bread wheat has got three genomes, A, B, and D. And when you cross things together that have different numbers of chromosomes, it causes a lot of mismatching, mispairing of chromosomes, resulting in sterility. So you couldn't cross these directly with wheat and expect to get fertile seed without a lot of serious intervention. So one thing that is done is to initially cross this diploid, this single genome plant, to a tetraploid, and then cross that hybrid to a hexaploid. Another thing that is done is to cross this diploid D-genome species directly to wheat and then do what's called an embryo rescue. So about a week or 10 days after fertilization, to go and dissect the growing embryo, extract it, put it under tissue culture conditions, and to try to get a viable plant. So it's not easy, but it is possible to introgress or to incorporate some of the diversity here into bread wheat, and that's what we hope to do.